All right, thanks, Eric. I'm Chris Hartle. I've been at the GSA group since about August of 2009. And I'm here to talk a bit about analyzing variant calls that you've made through our pipeline or through some other pipeline to assess them for how accurate they are or how they differ from one another, or to assess particular subsets for maybe you want SNPs of high impact, maybe you want SNPs that look interesting for some other way. So here's sort of where we stand. You've gotten all of your BAM files, collected your data, sequenced them, you have recalibrated them, you've reduced them and made calls in a large cohort, perhaps with other samples to better inform our understanding of the allele frequencies using our population genetics modeling. And you come down all the way to having a VCF. The pointer is not, here it is. Yeah, to having a VCF that's been completely genotype refined and it's been annotated. And the question is, how do you manipulate this file using the GATK? What kinds of things you can do? And this is sort of a large field, but the general idea, or the general way I tend to think about VCFs is a big matrix, where you've got rows that are the particular sites of the genome, the particular variants, and the columns are samples. And what you want to do is operate over this VCF by either subsetting sites, subsetting samples, or aggregating information in either direction or in both directions at the same time. And the tools we use to do these things are combine variants, where you're trying to add rows or columns, select variants, where you're trying to select out columns, select out rows, and variant eval, where you're aggregating information across this whole big matrix. All right, so a bit of background here, to take a step back, is that we want to answer biological questions here. The whole point of doing the sequencing and calling was not just to get a set of SNPs. It was to answer some experiment that you've done. Maybe it's a case control study. Maybe you just want to characterize the distribution of a particular type of variant in a new species. And so you want to answer these questions given this observation of genetic data, and you want some sort of sense of the quality of the data before you go off and make inferences. And so in order to do that, you need to be able to query and evaluate your call sets. And so what I'm going to show you, there are many ways of doing this, but I'm going to show you some real recent examples of having done this in the GATK. All right, so as I was mentioning before, when you're doing an analysis, you typically are doing it over a subset of calls. So for instance, if you're a clinician and you have a rare disease that's affecting a few children in the area, typically what you do is you sequence them, and then you say, I want to exclude every single variant that has already been seen because I know these are not causing the disease. And so you want to subset just the variants that are unique. Or if you're looking at population differences, you want to see the variants that are varying in allele frequency between populations. And so a lot of these analyses have to do with subsetting or combining different sets of calls. And so one way of subsetting your calls is through our tool called select variants. And this takes the original VCF file, which is what you want to select out of, an expression. So for instance, you want to select allele frequency less than 1%. You're interested in rare variants. So you can use that through the select expression. And that will select out rows based upon what's in the info field. Additionally, you can select out columns by giving sample IDs or expressions that will match certain sample IDs. And that will help you extract out columns. There are other things that you can do to manipulate the VCF. You might say, I don't want to see filtered sites at all. And that's this flag. You can say, after I've subset my samples down, some sites will be completely monomorphic in that subset of samples. I don't want to see them. That's that one. And then for all of these selection criterion, there are exclusion criterion. So you just reverse it. Rather than saying, I want to see all the variants less than 1%, you can say, I want to exclude all the variants less than 1%. And you do that by prepending this XL. And then you can select a particular type of record. So you want SNP or you want an indel. Right? And then you just give it a simple output, the VCF. All right, so one question that I recently asked was, how many variants did we happen to have in the European cohort of 1,000 genomes? I wanted to know basically the amount of heterozygosity that you find in Europe and contrast it with what we're finding in particularly samples from Sweden. And so here's how you do it, right? Select variants, here's the VCF, and I'm giving this sample file, right? I'm selecting just columns. I've here, this lists all of the European samples by their sample IDs in a column, right, just delimited by new lines. I'm going to exclude all of the variants that are suspect, and I'm going to exclude all of the variants that are monomorphic in Europeans. So variants that are unique to Africa or unique to Asia will not appear in this output VCF here, right? And so what I get out of having run this command line are only non-QC fail sites, so those are the rows, that are polymorphic in at least one of the European samples. And then I can just do a quick word count on the file to determine how many SNPs are present in the 1,000 Genomes Project European samples. Now, in addition to wanting to contract this big matrix that is your VCF, you might want to expand it. You might have done 2 different sequencing projects, or you might want to include information from the 1,000 Genomes Project, and you want to expand the number of samples or expand the number of sites. And this you can do with combined variants. And in its simplest form, it's quite easy. You provide, as multiple arguments to this -v, all of the VCFs, all of the call sets that you want to put together, and say, stick it in this output VCF. And it's smart enough to sort the files appropriately, and if there are samples present in one call set and not another, they'll get no calls for the sites at which they hadn't been assayed and things like that. Now, there are other parameters that basically take over when there are more complicated situations. So for instance, you might have call sets where at a particular site, in call set A it was filtered, in call set B it was pass filter, and you can ask, what do I do with those filters? Do I say that's filtered, or do I say that's passing filter, right? Or do I just say in one it's just treated as it's not even called, right? And what do you do if you have overlapping samples? Then you have how do you merge genotypes together, and what priority do you do so? This is all on the wiki, or rather all on the GATK docs, and it can get a little bit complicated depending on the complicated structure of your particular experiment. But in the simplest case, it's just provide what you want to put together and tell it where to stick that information. All right, so now let's say I want to actually combine. I want to see what sites are potentially unique to my other cohort here. So I have this 1,000 Genomes European dataset that I just generated using select variants from the full 1,000 Genomes project. And I want to say, how many more variants do I get if I include this second cohort? I'm just going to use combined variants for this. We have a tool. And again, it's just 2 -v's and a -o. And I say, anything that's been filtered, I don't even want to see. Pretend it's not even called. And then what I get is an output VCF that has all of the called, not filtered sites that come from one or both of these cohorts. In addition, combined variants automatically puts in a set key in the info field. And this key will tell you which of these 2 cohorts that particular site has come from. So it will say set equals cohort 1, set equals cohort 2, or set equals intersection for it's in both. And then you could, for instance, filter on that key via select variants again to find those sites that are unique to one in particular. All right, so here is a real example. One of my office mates, in fact, was putting a presentation together for ASHG, and she was interested in, for a family that had been sequenced for type 2 diabetes, which variants were private to that family. She wanted just to analyze those private variants. And she'd already extracted the sites that weren't in 1,000 genomes, and she'd written up a whole Perl script to do this. And she was tasked with using additional information from the exome sequencing project. And she didn't want to have to rewrite her Perl script to take in multiple things. And she asked me, what would you do? Is there a tool in the GATK for this? Well, there is. It's just 2 command lines. First, you want to combine them, and then you want to select for things that have only come from this one call set. So your first step is just the combined variants that we ran previously, right? You want to generate your new VCF that have sites from your exome sequencing project, from the 1,000 genomes, and from this type 2 diabetes sequencing study, right? And so you pass in these 3 -v's and this 1 -o. And this gives you this set information on the output VCF. And in the second step, you use that set information and just extract those that came only from this particular input, right? And now you have the set of variants in this output VCF that are private only to this family that had been sequenced for type 2 diabetes. And this went directly into a presentation just characterizing the allele frequency and the occurrence and the Mendelian violations in this particular set of SNPs. So this is a real question that the GATK just sort of solves for you with 2 short command lines. All right, so that's sort of how to put together call sets and to subset a call set that you do have. But how do you assess a call set? And so way back in the day, the way this was done was we had some expectations from population genetics. We had some expectations based on the divergence from the human genome and the chimp genome. In today's world, where you have a proliferation of sequencing data and you have databases like dbSNP, it suffices just to compare your data sets with the gold standard. And so what I'm going to be doing here is a real analysis I did. There were 62 samples from Northern Europe, in fact, from Sweden. And I wanted to assess how good this call set happened to be. And so, it happened to be low-pass sequencing. So the 1,000 Genomes project seemed like a good point of comparison. And so for these 62 samples that I had that were sequenced on an unknown technology and called by some unknown process, I'm comparing them to the 1,000 Genomes finished samples. So I've matched their basic geographic location to take care of any big population stratification differences. And I just want to compare them to see if their metrics sort of line up. And I'm going to do this using our tool called variant eval. And so variant eval takes in multiple VCFs via this eval argument. And what it'll do is aggregate over each of these VCFs particular attributes. So for instance, the number of SNPs, the transition transversion rate, the length distribution of indels, the number of structural variants, a whole host of evaluators. In addition, you can use as comparison any number of VCFs. So you can compare to dbSNP, you can compare to genotype chip that you ran, you can compare to any other call set that you can imagine. And we have a special case for just the dbSNP track if you want to say, break things down by present or absent in dbSNP. This EV argument allows you to select evaluation modules or evaluators, and ST allows you to select stratifications. So you might want to stratify based on how you filtered, based on a functional annotation, based on per sample versus in the aggregate. And that's what ST allows you to do. And then the output is a big variant eval table, which should be on the next slide. So this table has been sort of optimized to be read in via scripts that we have in the GATK. And so for each, so this file consists of a set of these tables just concatenated together. And it starts with a header and a parsing format. And then it gives you the names of all of the columns. So in this one, we have comp overlap is the name of this particular evaluator. And it's telling you how much a particular input call set is overlapping comparison call sets. So here are the evals, again, passed in through this eval argument. So I have the calls that I'm making, my test calls, and the 1,000 Genomes finished samples. And there are the comparisons, so the 1,000 genomes, all of them, and then the Omni chip, which is a genotyping chip, just those sites that are monomorphic. And it tells you the number of variants that were in the eval, the number that were novel, and how many of these eval variants overlapped variants of the comp. And so that will give you something like the dbSNP rate. So for instance, one thing you can read off right here is that at sites that are known not to be polymorphic, I have made 9 calls. All right, so here's how I evaluated that call set. In fact, I ran variant eval. I gave the calls I wanted to test. I wanted to compare to the 1,000 genomes and these particular 62 finished samples. And I compared against known polymorphic sites, known monomorphic sites, and all of the 1,000 genome sites. And I gave it as to stratify based on novelty, dbSNP 129. Okay. I went through that already. And so what I get is this big output table that you saw before. And you can condense it down and say, here are the known SNPs for my calls against the finish and novel. And you can already see a little bit that there's a slight difference in the novel calls. That somehow there are about 30,000 calls that are unique to my call that are that I would expect, 30,000 more that I would expect that I'm not really seeing. But all of the other metrics very much line up, right? And so what I've done here is I've taken a gold standard data set, 1,000 genomes, matched samples in size and location to my own call set that I wanted to evaluate, and I looked at a number of metrics that came out of variant eval. And I looked for differences, and one popped out. It's a little bit worrisome. And so what I'm going to do now is I'm going to say, if this is particularly an issue, it could be overfiltering, or the samples could be contaminated, or we could potentially have related samples in some way. And you might imagine that if we had overfiltering, right, then you would expect a lower number of SNPs per sample to fall into this novel set. So all I'm going to do here is add in this stratification by sample, and all I'm going to do is count the variants and do some basic other things, right? And so if you stratify them by sample, you get an output table which has an additional column that just is the sample ID, and stratify by the number of novel variants. You see a very big difference between the test calls that I wanted to evaluate and what you see in the 1,000 genomes finished samples. And this is due probably to overfiltering or relatedness. But you can see that there is a slight bit of suspicion in the novel sensitivity for these samples for whatever reason. So that's sort of a whirlwind tour through sort of the workhorse methods of variant analysis and evaluation in QC. And all of this is sort of complicated stuff because there's a lot of potential analyses you want to do, and we try to make them all available to you. So what I've shown you is just a very stripped down subset of what you can do with these tools. So please look at the GATK docs, play around with these for a while. They'll really help you. Are there any questions? Thank you.